Hi, my name is Shantae Stewart, and we are going to talk about the cytoskeleton today. So, this is a picture of the cytoskeleton broken down. This is the microfilament. So, this is the actin, and this is the intermediate, and this is the microtubule. So, the cytoskeleton, it helps itself to maintain its cell shape, and it acts as a force sensing device. So, it's also known as a mechanosensor. Three types of the cytoskeleton filaments are prevalent to many eukaryotic cells and are essential for spatial organization of these cells. So the intermediate filaments will provide strength mechanically and resistance to any types of stress. Microtubules will control the position of the membrane enclosed organelles, and it will direct any intracellular transport. And lastly, the actin filaments, they regulate the shape of the actual cell surface and they are needed for complete cell locomotion. So, going into actin filaments. So, here is um, basically a picture of what you would see on the microscope. And this is will be the breakdown of the G actin to come up fully with the F actin. So actin filaments, which would be the F-actin, they're linear, like large molecules or polymers of globular actin, which would be the G-actin, the subunits, and they transpire as microfilaments in the cytoskeleton as thin filaments, which are part of the contracting apparatus in the muscle and non-muscle cells. They normally trigger the plasma membrane and are typically collected at the cell periphery from any adhesion or membrane extension sites. They're able to make a number of linear bundles, so any two-dimensional systems or three-dimensional gels. And when actin will bind to these proteins, they're able to impact by making a precise structure and the filaments will shape. Actin filaments are mainly they're rich underneath being a part of the plasma membrane at the location, and they make a network that provides reinforcement of any determining cell shape, and it will permit the movement of the cell surface in addition to enabling cells to migrate, engulf particles, or divide. So, talking about the subunits, G actin has tightly binding sites that intervene from the head to the tail interactions with two other actin monomers. So, actin monomers normally combine to form a polymer to make the filament of the F actin. So, for each actin, the nucleotide is ATP or ADP rather than a GTP or a GDP. So, to get more talking about the polarity is next. The polarity of the actin filaments are envisioned by embellishing the filaments with globular heads as far as Si. They'll be cleaved off by myosin by the proteases. Bound myosin heads cause a appearance of arrowheads when you look in a electric micrograph. So next would be the microtubules we'll talk about. And for the microtubules, they're also filaments, so they're known for transcription of genes and coding for the tubulin, which will manufacture the messenger RNA. Then it'll translate from the messenger RNA by the means of the ribosomes for the production of protein. And the cells have two types of tubulin, which is alpha and beta tubulin, which are also known as a heterodimer. They come together. Each alpha or beta monomer has a binding site for one molecule of GTP. The GTP that's bound to the alpha tubulin monomer is confined at a dimer interface and it won't be hydrolyzed. So that's giving insight to the heterodimer being an important part of the structure. The nucleotide on the beta tubulin will be in a GTP or a GDP form. That's making it able to be exchangeable. The GTP cap will also be reserved by the microtubule as long as a GTP bound tubulin molecule is being added faster than the GTP is being hydrolyzed. The rate of this polymerization will slow down, and the GTP bound to the tubulin at the plus ends of the microtubule will also be hydrolyzed to a GDP. When this process occurs, the GDP bound tubulin will separate, causing a rapid depolymerization, and it will shorten the microtubule. So, next we will talk about the intermediate filaments. So, they appear to play a structural role by giving strength for the cells and tissues. So, intermediate filaments are also connected in the creation of the nuclear lamina. So, that will be underneath the lamins. It's a net like meshwork array that lines the inner nuclear membrane and governs the shape of the nucleus. The nuclear lamins, which establish the fibrous protein network, lines the inside of the nuclear membrane. So, due to its close vicinity to the membrane, the nuclear lamins help attach the chromosomes to the nuclear membrane and provide anchorage points for the nuclear pores. And lastly, we're going to talk about cell locomotion. So, cell locomotion deals with polymerization, rearrangement, keratin movement, and myosins. So, first, we're going to talk about the picture that will be known as the keratin movement, that which we'll explain later. So, from the process of association for the actin filaments, with the development of actin filament bundles, along with the networks to the contracting packs of actin and myosin, and the moving myosin molecules on a single actin filament. All of these mechanisms are basically characterized by the main methods at the location of the cells to produce actual migration. So polymerization that occurs on the actin filaments at the location of the leading edge and the crosslink of the filaments to form bundles in their networks. So proteins that would happen to be part of this process would be AARP, and it's 2/3 in VASP, which will aid in assembly. Rearrangement, this occurs on the actin filaments during a keratinocyte movement. So a keratinocyte movement is when cells transfer by the method of crawling over surfaces instead of cilia or flagella to swim. And the nomad will crawl nonstop looking for food. They will be seen either attacking or consuming smaller ciliates or flagellates in a small like pond water. And so normally in animals, it's almost all cell locomotion occurs by crawling. So you can see the different steps as far as in the picture of how the anemobae would be crawling. As far as myosins, there's two types. Myosin 1 is part of the extension or forward motion of the cell, so that would be the crawling. And myosin 2 is the retraction or the withdrawal of the cell body, so that's giving the role of the translocation part. So that would be it picking itself back up to move, step forward again.